Hello everyone, a very warm welcome to all of you. The topic for today's discussion is mammalian fertilization. We will study the process of fertilization in mammals. As we all know, fertilization is defined as the process where we get to see a sperm fusing or uniting with the egg to give rise to a diploid zygote, which then develop into a new individual. This is what is happening in fertilization. Now, how is it happening? Where is it happening? All these details are going to be addressed here in this lecture. Okay, so the mammalian sperm that fertilize the egg must reside in the female reproductive tract before they are able to undergo the acrosome reaction. Now, this process or this maturation process is called capacitation. We will learn about all these things today in detail. The mammalian egg is surrounded by an extracellular envelope, which is called the zona pellucida, to which the sperm must bind and penetrate before they can make contact with the surface of the egg itself. So these events which are seen in mammalian fertilization are all summarized here, or we will be discussing that here. So let's get started here. So what happens, see, the sperm is activated in the female reproductive tract itself. So the female reproductive tract activates the sperm. Then what happens? Sperm binds to zona pellucida. Then what is happening? Acrosomal reaction, and finally one sperm, the winner or the champion, finally lyses a hole in the zona. So this is what is happening. In this diagram here, you can see the zona pellucida, which we are talking about. The zona pellucida has the follicle cells at its outer surface. The sperm has to digest everything to get an access inside the egg cell. Now, how does it gain entry into the egg cell? If you take a note at these three layers, 1, 2, and 3, the first one is the cumulus layer, the extracellular coat of the egg. Then we get the zona pellucida, which is the extracellular matrix, and finally the egg cell membrane. The sperm comes and gets associated with this, with the first one, the cumulus layer. Then it travels little deeper. Acrosome reaction happens here, and finally it gets an access to the cell membrane, which is digested, and the sperm goes inside to fuse with the egg nucleus. Now, where is it happening? What is happening? We should know. See, the narrow region of the oviduct. The narrow region of the oviduct is called the isthmus. Isthmus. Now, this isthmus plays a very important role in preventing polyspermy. The swollen region of the oviduct is known as the ampulla. Now, this ampulla is the site of fertilization. This is the place where fertilization occurs. Ampulla is the site of fertilization. The edges of the fimbriae receives the edges of the fimbriae, they receive the oocyte cumulus complex. Okay, now through muscle contraction and ciliar motion, the oocyte is sent to the ampullary region. The oocyte must come to the ampulla, the ampullary region. Once the ampulla, the structure is somewhat like this. Here, I'm just making a diagrammatic representation. Here it should be a little wider. Then here it should come. The egg should come to the ampullary region. And it waits for one sperm to come and fertilize it. Now, who is sending this egg here? Muscle contraction and ciliar motion of the oocyte. Ciliar motion sends the oocyte to the ampullary region. Now, experimentally, if the matrix of the oocyte is removed, then what will happen? Then fimbriae cannot bind to the oocyte. So this is what is going to happen if experimentally the matrix of the oocyte is removed. Okay, now we'll come to the process that help in sperm translocation. Just now we have talked about how the egg comes to the ampullary region. Now we must know how the sperm should also go. This is not one-way work. The mother as well as the father both have to work equally, as they should do after the child is born. Anyway. So what happens here? What plays an important role in sperm translocation? See. First is sperm motility, uterine muscle contraction, and sperm rheotaxis. Clear? Now, what are these? Sperm motility is the capacity or the ability of the sperm to swim, to move. Sperm motility has come from it has come from the concept how fast the sperm can travel towards the egg. Uterine contraction is how well the uterine muscles are contracting. That also has some role in pushing or taking the sperm towards its destination. And last, we talk about sperm rheotaxis. What does that say? Calcium ion channels open in the sperm and cause calcium influx in it, which helps in sperm movement. So, the questions can come, what is the main ion that is associated with this? Your answer is calcium ion. So, there is influx of calcium ion that helps in sperm movement to the oviduct, and this directional cue is provided by calcium ion. And another thing, remember, Casper genes get activated, that opens the calcium channel. So, what is opening the calcium channel? Casper gene. So, these two things you must keep in mind. Next, we will talk about sperm capacitation. What is it all about? So, in very simple, basic word, sperm capacitation is a process that activates the sperm. It is a very important process without which there is no fertilization. Okay, so what is that? The epithelial cells of the isthmus bind to the sperm. All right, the epithelial cells of the isthmus, mind it. They will first bind to the sperm, and then we get to see the whole process of capacitation. What happens? It is, now remember, capacitation is a series of physiological events. It is basically a physiological maturation event that takes place in the vaginal tract, uterus, and oviduct. All these are involved here. It can be accomplished in vitro for many species using oviduct fluid, culture media, albumin protein, and all these stuff. Capacitation involves changes in membrane, lipid, then carbohydrate, protein, membrane potential. See, protein phosphorylation, internal pH, and enzyme activation. This is very, very important, and all of these together play a very important role. Membrane potential means it becomes more and more negative. Always remember, capacitation is transient, and sperm become uncapacitated after a period of time. Now, what is happening? Capacitated sperm gets hyperactivated. They get rid of the epithelial binding and moves forward. If they are unable to get rid of the epithelial binding, they will not be able to travel forward. The uncapacitated sperm, they die in the epithelial binding because it cannot move forward, as it is unable to remove the binding, get rid of the binding. So this is very important. Now, another concept comes here, the albumin protein of the female reproductive tract also, like, it causes cholesterol efflux from the sperm membrane. The lipid tracts get disturbed, and calcium ion, potassium ion, and bicarbonate ion channels get open. This is the next part which we are going to study. See. Now, this is something which is happening in the female reproductive tract. Okay, this is what is happening in the females. The lipid rafts get disturbed and calcium, potassium, and bicarbonate channel opens. You should know what is going in and what is going out. Remember, calcium and bicarbonate ions get inside and potassium goes outside. Calcium and potassium doesn't follow the same direction. Calcium and bicarbonate ions, I'm again repeating, they come in and potassium goes out. Clear? Now this causes adenylate cyclase to convert ATP to cyclic AMP, which activates pKa. Then pKa phosphorylates tyrosine kinase, which in turn phosphorylates sperm protein like Zumo. This causes sperm capacitation. Now see how important calcium is. Calcium causes increased dynein activity, leading to hyperactivity of sperm, enabling the sperm to swim faster. Now see, we'll come across three important terms: hyperactivation, thermotaxis, and chemotaxis. What are these? See, hyperactivation is what? Motility pattern change in the oviduct in some species. Now, hyperactivated motility means like higher velocity, greater force suited for viscous oviduct fluid. So what's there? See, the oviduct fluid is viscous. Okay. Now, to swim through it, the sperm has to be a little more strong. It should have certain ability to overcome this resistance, which is offered by the viscous fluid of the oviduct. So, the sperm should have higher velocity and greater force. Higher velocity and greater force. This is called hyperactivation. What is thermotaxis? The sperm may be able to sense a thermal gradient. The ampulla of the oviduct is little more warmer than the isthmus, and only capacitated sperm can respond thermotactically. Remember, capacitated sperm can respond thermotactically. The incapacitated one won't be able to respond. Then what is chemotaxis? Chemotaxis is, remember, chemotaxis is the nearest range reaction, thermotaxis is moderate range, and rheotaxis is long range. The oocytes and cumulus cells may secrete chemotactic agents. Like follicular fluids show some chemotactic ability. Only fertilizable follicles have chemotactic activity. Only fertilizable follicles have chemotactic activity. All right. So progesterone and CRISP1 are chemotactic compounds. These two names you must know. Progesterone and CRISP1. These are chemotactic compounds and progesterone open calcium channel in sperm tail. This is also important. So they help in opening in sperm tail. Clear? So these are certain points which you will have to keep in mind. Certain important terms are there which often give rise to big questions. So don't underestimate anything. Now we'll talk a little about the egg. The mammalian egg, as you know, is surrounded by an extracellular envelope called zona pellucida, to which the sperm must bind and penetrate before they can make contact with the surface of the egg itself. The zona pellucida contains three glycoproteins called ZP1, 2, and 3. Okay. See? Zona pellucida has ZP1, ZP2, ZP3 that polymerize to form a gel. These glycoproteins actually, they polymerize to form a gel. The zona of newly ovulated eggs is surrounded by a constellation of follicle cells in a matrix of hyaluronic acid. So here, if you see, if I show you the picture, you'll understand. See, always relate pictures or diagrams to what you are studying. That is very important. Without this, you will not be able to remember the topic, the context. So always try to connect. See here, to save time, I will have to skip few diagrams, but when you are studying it, please don't skip anything. Study everything in detail, refer to the diagrams to each and every point as meticulously as possible. Now see, ZP2 interacts with acrosome reacted sperm, while ZP3 interacts with acrosome intact sperm. Acrosome intact means, acrosome intact implies what? You know, any of you? The acrosome reacted sperms are those sperms where exocytosis has already happened, and intact ones where exocytosis hasn't taken place. ZP3 has N-acetyl glucosamine residues on it, and their Receptors are present on intact acrosomes. The receptors are known as, the receptors are this. 1 second. These are the receptors for N-acetyl glucosamine. See, so what did I say just now? ZP3 has N-acetyl glucosamine residues on it, and the receptors are this GALT that are present on intact acrosome. Now, sperm galactosyl transferase binds to ZP3 N-acetyl glucosamine. Fine. The zonal layer gets sliced and fusion follows. This is the event. This is the flow of event. This cross-linking activates specific G protein in the sperm membrane, and it initiates a cascade that opens the membrane calcium channel and results in calcium-mediated exocytosis of the acrosomal vesicle. Now, isomo protein of sperm reacts with JUNO, Juno protein of egg microvilli. This leads to recruitment of CD9 to the area of egg and sperm addition, and this is followed by sperm-egg fusion. So, whatever I have said just now, I'm just writing it out for you in a simple flow chart. So, what did I say? ZP3 has N-acetyl glucosamine. N-acetyl glucosamine residues on their body, and receptor for this N-acetyl glucosamine is what? This. On intact acrosome. Clear? Sperm galactosyl transferase, I just now told you, sperm galactosyl transferase binds ZP3 N-acetyl glucosamine, and thereafter what is happening? The zonal layer gets sliced and fusion follows. Fusion follows. So this is what is happening. What will happen due to this cross-linking? This cross-linking will activate specific G proteins in the sperm membrane. Okay, and what will happen? It will initiate a cascade of events. It will open the membrane calcium channels, and it will result in calcium-mediated exocytosis of the acrosomal vesicle. Then what is going to happen? Isomo protein of sperm will react with which one? Juno of egg microvilli. This will lead to This will lead to recruitment. CD9. Clear? To the area of egg and sperm addition. Egg and sperm addition. And this is followed by egg sperm fusion. So this is the entire story of mammalian fertilization. And another topic is there which we are not going to discuss today. Just remember, in mammals, slow block to polyspermy occurs. In sea urchin, if you remember, we had talked about slow block as well as fast block. Here, slow block to polyspermy is seen to occur. So, this is all about our lecture today. Thank you so much for attending the lecture. Happy learning. Till then, goodbye.